I'm going to give a somewhat different talk than I usually give. We'll see how it goes. What I want to do today is talk about the idea of intrinsic motivation. And I'm going to talk about this in a variety of natural, meaning biological, but also engineered and hybrid systems. And if you're interested in the details, everything, the software, the data sets, the primary papers, everything is at this lab website. And then this here is my personal blog around some ideas of what I think all of this means. So when I say motivations, what I mean is large-scale causes of behavior across systems. And that takes many forms. For example, in biology, it often takes the form of asking why did the system do what it just did? And the explanations can range from biochemistry to psychoanalysis, depending on what kind of system you're talking about. And as engineers, we would like to know how to make systems do things. And so this is engineering, but also bioengineering, computer science, and so on. So this idea of why it is that systems do specific things and how we can communicate to those systems to do something else. So these and typically in computer science, when we think about where do the motivations of systems come from, typically we think about a designed algorithm. So a human or perhaps an AI designed a specific algorithm to do a specific thing. Sometimes it comes from learning. So there was a reward function and the system learned to do something. Sometimes there's an evolutionary algorithm, meaning that it was selected for a specific fitness function. And then, of course, there's also morphological computation and physics that can be exploited in robotics and engineering. In biology, some of these things overlap. We tend not to like this idea, this idea that there were designed algorithms, but the rest of this fits. Learning, of course, all kinds of living systems can learn. Obviously, evolution, so selection for specific functions, and a lot of emphasis on physics. So this is what I'm interested in, understanding whether these are sufficient and how can we think about motivations across a range of systems. And to help us think about this, I would like to propose a continuum. So I don't like binary categories, things like machines versus living organisms. I don't think those kinds of categories are useful. What instead I like is a continuum so that we can ask what exactly is changing, what is different across this continuum, and in particular, what kind of tools and approaches can we use to motivate and to understand the motivations of systems across the spectrum. So if you are only amenable to hardware rewiring, you might be down here. If you are a cybernetic kind of system that's amenable to control theory and the tools of cybernetics, maybe you're here. And then, of course, here we start seeing systems that are trainable by rewards and punishments, and then eventually systems that communicate via reasons and are amenable to psychology, friendship, love, and things like that. So the important thing about the spectrum is that where a system fits here is not obvious. It cannot be determined from a philosophical armchair. So you can't just say, well, this is a machine, therefore I know it doesn't have cognition, or this is a cell and I know it's not intelligent, only brains are intelligent. None of that is really scientific or useful in engineering. What we actually have to do is experiments. You have to take systems and find out which kinds of tools across the spectrum are actually applicable. And when you do that, you get many surprises. So here's the outline for today's talk. What I'm first going to do is to generalize the notion of patterns, which are forms of either structure or behavior, and many other things besides. And so I'm going to show you why I think these are actually the same thing. Then I'm going to show you a model system called morphogenesis. And so this is biology. So the first probably 2/3 of the talk are going to be largely around biology. And I'm going to show you a model system in which groups of cells solve problems as a collective intelligence. The best way to describe what they do is not some sort of mechanical machine metaphor, but literally a navigation of a problem space and a kind of collective intelligence to which many tools from cognitive science apply. So this is the model system that we do a lot of work on. And then we have to ask the question, if you have a system which attempts to fulfill specific goals, where do those goals come from? And so now we start talking about motivations, and I'm going to start us thinking about where goals come from that goes beyond selection. In biology, this means in genetics and the specificity of the environment. But what else is there? Where do motivations come from? And this will help us connect it to engineering and computer science. Towards the end, I'm going to argue that in order to understand where these motivations come from, and more importantly, to be able to predict and to call up the ones you want in your artificial systems, we have to start thinking about what you might call a Platonic space. It's a structured space of patterns, of form and behaviors that is really critical for both biology and physics, and I'm going to argue briefly that basically standard reductive physicalism is quite insufficient and for either causation or explanation in these systems, and more importantly for building, for creating new things. And I'm going to argue at the end too that even extremely simple interfaces are the benefits of some of these ingressing patterns from this latent space. And at the end, I'll just talk about our research program and what we are doing to study this space and the mapping between the patterns in that space and the physical interfaces that we make. So the first thing to remind us of is that while many textbooks and papers in the philosophy of mind and so on start off here as a modern human, an adult modern human, we all took the journey across different disciplines. So we started here as an unfertilized oocyte, and so this was considered the province of chemistry. Then there was this amazing process of self-assembly that gave rise to either something like this, which we recognize now is to be handled by behavioral science, or even something like this by psychology. But that's not the end of the line, and different components here can do multiple things afterwards, and they can take paths on the field of oncology by disconnecting from this collective, or even bioengineering, and I'll show you some of these anthrobots shortly. So what we need to understand is the pattern of scaling. How is it that the simple system here becomes something like this, right? There are no binary categories here. There's no magic lightning bolt at which point chemistry becomes cognition and so on. So the first thing that I want to get across is this idea that forms or patterns of body and mind are really part of the same thing. We need to think about them in the same fashion. And actually, Alan Turing was already on top of this, because here's someone who was very interested in intelligence, in reprogrammability, in embodying cognition in other substrates, and at the same time, he wrote this amazing paper on the chemical basis of morphogenesis. He wanted to know how chemicals in the embryo self-organize. Why would somebody interested in computing and intelligence be looking at chemicals in morphogenesis? I think it's because he recognized these are fundamentally the same problem. And I think this is a very deep point. So I'm going to show you a few examples. These are the kinds of things that are easy for us to recognize, right? So here, this little squirrel has a very good theory of mind. He knows his owners are going to be upset to find him in this accident scene. So there, he sets up this little scenario, and then if nobody comes to check on him, he's actually going to look to see has anybody noticed all the hard work he's put into this. So he's looking. Okay, these kinds of things are easy for us to recognize because they happen in the same problem space that we share, so three-dimensional space. They happen at the same time scale and so on. There are other things like this which sort of look like we understand what's going on, but it's not obvious at all, and we can't understand this from pure observation. We can't know. We have to do functional experiments to really understand what's happening. And so these are the kinds of things we mean by behavior. Now, even very minimal systems do this. So this is a single cell. So this is what we are all made of. Now, this is a free-living organism called the Lacrimaria, but all of us are made of single cells working together. There's no brain here. There's no nervous system, but you can see this incredible competency at solving its problems at the local scale. And if you're into soft body robotics or something like this, you're probably very jealous because we don't have anything that is this competent. And remarkably, even the material of which this creature is made, so molecular networks, right? So here, so molecules activating and suppressing each other's activity. The molecular components already have 6 different kinds of learning that they can do, including Pavlovian conditioning. So we are made of an agential material. There are competencies and agendas all the way down to the molecular level. It does not start at the brain or the nervous system, all the way into cells and the material of which the cells are made. So this is all kind of in the behavioral sphere. Now let's look at shape, anatomy. Well, the behavior of molecules underlies the shape of tiny life forms. So these are diatoms, and you can see this whole thing is one cell. It's extremely small, but this is one cell. These are all single cell creatures. They have very specific shapes that are defined by the behavior of molecules. But morphogenesis, so the generation of form, is an example of that, but on a much larger scale. So we become embodied through the action of many cells working together. If you take a cross-section through the human torso, you see this incredible order, right? So everything is in the right place, the right shape, the right size, next to the right thing. And if something is damaged or missing here, we need to know, where does this pattern actually come from? How do we convince these cells to rebuild this? And a large part of my lab does regenerative medicine, where we try to regrow organs and things like that. And of course, there are many amazing morphogenetic structures. For example, this fly is running a very simplified, stripped-down, two-dimensional ant morphogenesis program on its wings, and it does this to scare predators who don't want to deal with ants, and it wiggles the wings, and it looks like the ants are kind of running around. So there's a program here of making the shape of these ants, but also of the fly itself in 3 dimensions. And then there are morphogenetic events outside the body. For example, the collective of termites will make this incredible complex structure. Spiders will make this structure, the spider web. And what we have to understand is that none of these patterns are in the DNA. A lot of people, if we ask, where does this pattern actually come from, a lot of people think, well, it's in the DNA somehow. But the genome doesn't say anything about this any more than it says what the structure of the spider web or the termite colony should be. The genome contains the specification for the hardware, the micro level proteins that every cell gets to have. All of this arises as a function of the physiological dynamics and software that are running on this system. And so the reason I say that the forms of behavior and anatomy are the same thing is because they are underlined by a symmetry of this notion of a space. So what space do systems navigate? Well, we as humans are kind of obsessed with 3-dimensional space because of our own evolutionary history and our vision, the importance of vision, and so on. And so we can recognize navigation of spaces, meaning intelligent behavior. We can recognize navigation of medium-sized objects in medium speeds moving through 3-dimensional space, birds and mammals and especially primates and things like that. So we can recognize navigation of that space. But biology has been navigating all kinds of other spaces that are often high-dimensional, really hard for us to visualize, and not obvious at all. For example, the space of possible gene expression states. So this might be a 20,000 dimensional space. Or the space of physiological states, right? And in particular, one kind of behavior that we study in our group is morphogenesis, the navigation of systems through anatomical space, meaning the space of possibilities. So you can think about the movement of the configuration of the body from a single cell towards the shape of a human or a snake or a giraffe or a tree or whatever you're going to be, as a kind of navigation of this anatomical space. So all of these, so morphogenesis, the generation of the body shape, becomes just a kind of behavior, albeit in a different space at a slightly different time scale. Now, I'll point out that deciding what is morphogenesis and what's behavior is actually really hard. So here is something interesting. This is a slime mold called Physarum polycephalum. It's growing out. Here's one glass disc. Here are 3 glass discs. What you'll find out is that during this time, while it's growing evenly, it's gathering information about its environment by gently pulling on the substrate and reading the strain. So this is biomechanical sensing. It reads the strain angle, and then eventually it figures out where the bigger mass is, and boom, and it goes right, reliably, it goes right to the bigger mass. And it can do this partially because it has these, it's basically a kind of a hydraulic computer. It has these flows inside with independently addressable synapses here in between all these structures. But is this behavior or is this morphogenesis? Well, it's both, and trying to label it is really not helpful, because the way that this system exerts its behavior is through changing its body shape. It's through morphogenesis. This is all one cell. This is all one thing growing out into that. That is how it does behavior. So I think what happens is that evolution pivots the same tricks through different problem spaces. So very simple early forms of life were navigating metabolic state spaces and then physiological state spaces, and then the genes came along and the space of transcriptional states. And then multicellularity came along and we could navigate anatomical morphospace. And then nerves and muscles came along and we were able to navigate 3-dimensional spaces, and now linguistic spaces and financial spaces and who knows what else, right? So So the idea is that these spaces are really they're plentiful but also the distinction between them is really in the eye of the observer. That is, we as scientists decided to cut it up this way, but as you see from the Physarum, it's not obvious that there is any kind of ground truth about what the space really is. And very importantly, with these kinds of systems, you have to ask, what do they see? What space do they think they're navigating? Okay, so what I've tried to do here is to generalize this notion of a pattern, and it's a pattern in any of these spaces. So it might be a physiological pattern, it might be a gene expression pattern, a behavioral pattern, an anatomical pattern. These are all the same things. Now, the next part of the argument is to point out that these patterns are not just emergent outcomes. It is, at least in biology, it is not the case that, like for example, in the cellular automata, there are local rules. You sort of turn the crank, execute the local rules, and then something complex eventually pops out. That certainly happens, but that is not the whole story, because these patterns actually function as goals, and you can see us sort of working up to this idea of motivations. The patterns are targets of behavior because they are goals that are being pursued, and they're being pursued by diverse means, and the degree of diversity and ingenuity by which different systems pursue specific goal patterns is basically a measure of their intelligence. In fact, that's one way to define intelligence, is how good are you at meeting the same goal by different means when situation changes. And in particular, I've defined this concept called a cognitive light cone, which is simply the idea that we can estimate the size of the biggest goal that a system can try to implement. So the size, in this case in space and time, but it can be in any of those spaces that I mentioned, the size in space and time of the biggest pattern that the system is capable of storing as a set point for its behavior, and trying to achieve that behavior. And so you can see different kinds of creatures can have different size cognitive light cones. Again, this is not the range of your perception. This is not the range of your possible actions. This is the size of the goals that you're able to pursue. And the idea is that goals are subsets of a motivational strategy, and goals are kind of what hold organisms or agents in general together. We see a collective and we say this is a goal-seeking system to the extent that all of its parts are aligned to meet a specific goal. And the interesting thing about biology is that the cognitive light cones are plastic. They can shrink and they can grow. So a single cell has a tiny little cognitive light cone. All it cares about is for scalar values of things like pH, hunger level, and so on, at a very small region of space-time, only a few minutes forward, anticipation and memory back, tiny little cognitive light cone. But when cells join into networks, and these are the kinds of, for example, multicellular tissues in your body, then suddenly they're capable of representing enormous, grandiose goals, for example, such as how to build a limb. And I'll talk about this momentarily as to why this is a goal. So the idea is that the goals can grow. So during evolution and during embryonic development, systems are able to acquire bigger and bigger patterns as goals, and they can project them into new problem spaces, and in fact open new problem spaces. And so now it's time for me to explain how this navigation works. So the basic fact of morphogenesis is that we start with a single cell, it divides, so that's the egg, it divides, and then eventually you get something like this. Now, this process is extremely reliable, and there's a huge increase of complexity from here to here. But those two things are not why I'm using agentic terminology. This is very important. Many times people will see something complex like this and they say, "Oh, wow, that's amazing, intelligence and all that." That is you can't say it's intelligence simply because it's reliable or because it's increased in complexity. Those are cheap. It's very easy to get those from feed-forward, purely open-loop kind of processes. That is not what's happening here. This is not pure emergence. The reason that I'm using this agentic terminology is that if you actually poke this process, if you do perturbative experiments, you find out something very interesting beyond this reliable thing. So first of all, if you chop embryos into pieces, you don't get half bodies. You get normal monozygotic twins, triplets, and so on. And so these systems are able to get to the same ensemble of goal states, a correct human target morphology, from different starting positions. You can start as an embryo cut in half. You can start as a normal embryo and various other things. And you can avoid local maxima and still get to where you're going. So that's interesting. You can get to the right place from different starting positions. In fact, something like this. We discovered this about 10 years ago. If you these tadpoles that have to become frogs normally, so here are the eyes, here are the nostrils, the brain, the mouth, like all of these things have to move to become to create a frog face during metamorphosis. And people thought this was a hardwired process. Every set of cells just moves in the right direction, the right amount, and you get a normal frog. Well, so we created these so-called Picasso tadpoles where we scrambled all the starting configurations. So the eyes on the back of the head, the mouth is over here off to the side. And yet these things become normal frogs too, because the organs don't just move in the correct direction, the correct amount. They actually they do a kind of error minimization scheme. They keep moving through novel paths until they get the right final shape. So this is a completely different process than simply feed-forward open-loop emergence. And more broadly, it's you can see this in the kind of process called regeneration. So this is an axolotl. It's an amphibian that can regenerate its limbs, its eyes, its jaws, and it's amazing. And what happens is that if the limb is amputated anywhere along the length here, the cells will very quickly become activated. They grow. They produce the original structure, and then they stop. That's the most amazing thing about this process, is that it knows when to stop. When does it stop? Well, it stops when the correct axolotl limb has been produced. So this is, again, a kind of homeostatic system. It has feedback. This simple model of open loop emergence is not a good fit for these data. You have to use cybernetic models with feedback. So this kind of anatomical homeostasis, where there is a set point, and as long as you deviate from that set point by more than some acceptable small error, the system will work really hard to correct it. Now, one thing that you'll note is that it's got the right number of fingers, the right shape, but no individual cell knows what a finger is or how many fingers you're supposed to have, but the collective does, the group does. And this is very important, that these are, the whole point about being a large-scale goal-seeking system is that you have goals that your parts don't have. You can align your parts towards goals that they don't know anything about. At each level, there are novel cognitive content being generated. And so what's important here is that what I just showed you is not about repair of damage. It's not a simple kind of in-painting where you simply kind of repair a static structure. That's not the key here. The key is this kind of top-down control. And I want to show you this experiment that was done back in the 1950s, where they took tails and they grafted them to the side of this amphibian. And what you'll see is that the tail slowly turns into a limb. Now, notice the cells up here at the tip of the tail. These are tail tip cells sitting at the end of a tail. There's no damage there. There's nothing wrong there. There's no injury. Why are they turning into fingers? They're turning into fingers because the large-scale system, which does in fact have an error, the large-scale system can tell that this is not, tails don't belong in the middle of this body. What it does is propagate that error and propagate the stress and the fact that the situation is not what it should be, propagates it all the way down to the lowest level components, so that even though the molecules, and meaning the molecular networks inside the cells and the cells themselves, they would have no idea that anything is wrong here, because in their local scale everything's fine, but they're being told by the higher level system that actually it's not fine and you need to become these fingers. So what we're looking here at is a multi-scale system where local order obeys a global plan. And this is, of course, the hallmark of a cognitive system. You do this too in the morning when you wake up and you have very abstract educational goals and financial goals and social goals and whatever. In order for you to get up out of bed and do those things, the chemistry of your muscle cells has to change, right? The potassium ions and whatever have to cross the muscle cell membranes in order for you to do voluntary motion. So your whole body is this transduction apparatus that takes very high level abstract goals and transduces it into the aligned actions of your parts of in fact very small molecular parts. So we're now seeing that these kinds of forms that we are talking about, the shapes of behavior of cell groups, are a kind of target for goal-directed behavior. Now, like any complex cognitive system, these kind of persuadable systems should be reprogrammable. And so I want to show you just one example, and I could give several hours talk just on this stuff. So I'll go very quickly through this. This is a planarian, a flatworm, and the amazing thing about these planaria are that if you cut them into pieces, and you can do it in hundreds of pieces actually, every piece will regenerate into a perfect little worm, and it's normally 100% reliable. But what we were able to do, because we learned to read and write the representation of these goal states within tissue. It happens to be bioelectrical. I'll show you a brief example of that. It happens to be just like in the brain, it's stored in an electrical network, but we can now see those patterns and we can change them. And so we could go in and say, no, actually, you should have two heads as a planarian. And then when you cut those animals, sure enough, they make two-headed worms. And when you recut those, they continue to make two-headed worms. And this is not Photoshop, it's not AI. These are real animals. You can see them here. The system will continue to remember the new goal and to pursue it under future rounds of regeneration. So again, these patterns are, they serve as targets of goal-directed activity, and they are rewritable. They are plastic. Not only can we change the number of heads, we can actually change the species type of head that you get. So this species that has normally a nice triangular head, we can ask them to make flat heads like a P. felina or round heads like an S. mediterranea, about 100 to 150 million years distance between these creatures and this one. And so what you see is that the hardware here is perfectly willing to visit attractors that belong to other species in this morphospace if their decision-making process is perturbed, and again, that's bioelectric signaling is how they navigate this space. Keep in mind the hardware is stock, we haven't touched the DNA. We haven't, in none of these cases do we change anything about the genome. This just illustrates how evolution makes these amazing problem-solving agents that are very plastic, and their goals are rewritable, and not just the shape of the head, but even the shape of the brain and the distribution of stem cells becomes just like these other species. Okay. So now what I've shown you now is that these goal states are rewritable, but in all of these cases, the goal states were natural products of evolution. In other words, yeah, we can make these weird shaped heads, but that's because there are other species that already have those heads. So all you're doing is walking around in this morphospace. But so now let's talk about what the novelty here can be. Beyond the forms that have already been selected for, what else is possible? And the key is that the hardware is actually able to find some very novel forms. Well, first, an evolutionary example in plants. So, you look at an acorn and it very reliably makes these oak leaves, and you think, okay, well, that's what the genome knows how to do. This genome specifies, it encodes this particular shape, and that's really not a good way to think about it at all. And we are taught this by a non-human bioengineer. So along comes this little wasp, and the little wasp does some bioprompting, which is to put down some chemical signals that cause these cells to build something completely different. This is not built by the insect cells. This is built by the plant cells. Okay, so these are called galls, and you see this amazing structure. Who would have known? We would have no idea that these flat green cells are capable of building something like this if this plant didn't hack them into a new region of morphospace. And remember, it doesn't do this the way that it builds its nest, which is like a 3D printer to go along and just lay down the structure and build this nest. That isn't what happens. It puts down some signals, and then it basically motivates the plant cells to build something very different. But so the standard evolutionary default, the flat green leaf, is a point. It's a single point in that latent space. But we now see that, okay, there are other interesting outcomes besides that point that we can reach. And I just want to show you a couple of cute examples. Wait, let me pause this video, because along with hacking other systems, your first job as a high-level collective is to hack your own parts. You have to have control over your own components and get them to do new things. So I want to show you a couple of examples of biology. This is that flatworm that I showed you. This is a planarian. And it has a little tube that it can put out called a pharynx, and this is how it eats. So it can suck up nutrients using this little tube. But normally you don't see this. This stays inside the body, and you almost never see it. But if you take it away from the rest of the body, if you liberate it from the rest of the body, turns out that these little pharynxes, here's one, they have their own life, and they live for hours, and they run around. And this one in particular is going to attack this piece of liver, and it's going to actually burrow through it because it doesn't realize that there's nothing on the back of it. So as it eats the liver falls out the other side. But you can see it come out the other way. The pharynx actually has lots of autonomous activity, and being a normal planarian involves 99% of the time keeping that activity suppressed, keeping it inside the body, keeping it from running around and doing things on its own. Here's another example of what it means to hack a biological system. So these are cells, and I'm going to play this video in a moment. You're going to see the colors represent voltage. So the voltage potential across the cell membrane. So this cell has 1 voltage, it's blue. These cells have another voltage, it's green. And so this cell is crawling along, minding its own business. Here it goes. Now the green cells are going to reach out and they're going to touch it, and boom, that's it. That's all it takes. That tiny little touch is all it takes to turn that cell green, and now it's joined the collective and it's going to be part of building whatever these things are building. So this is the kind of thing that cells do to hack each other's behavior. And it's very important for us to learn how to do this because we would like to control what cells build. And here's an example of that from our regenerative medicine program, where we take an early frog embryo and we provide a bioelectric signal to a group of cells that basically tells them to become an eye. And if we do this in cells that are going to be a gut, here we go. Sure enough, they build this eye. So we want to be as convincing as possible so that we can build new organs for regeneration, for transplantation. And you can see here, this is a cross section of this early region. The blue cells are the ones that we injected. So we want them to acquire a new goal, to form an eye. We want them to convince their neighbors, because there's not enough of them to make an eye, so they have to, all these other cells that are not blue, we didn't touch those. The blue cells convince them to make an eye, right? So there's a second level of instruction here. By the way, these other cells resist, because it's a cancer suppression mechanism. If somebody's telling you to become a weird voltage, you try to resist and in fact normalize them out. And so they resist, and there's this like battle of worldviews that happens where these cells are saying we're going to build an eye, and these are saying no, you should be gut cells, and depending on who wins, you either get an organ or you don't. And this is the kind of hacking that we want to control. Again, none of this is manipulating DNA. None of this is even seeking to manipulate specific gene expression. It's about convincing a complex system to undertake a new goal. So again, this issue of motivation, right, of adjusting their motivation, take on a new goal, and also to take on the hard part of coordinating all the downstream steps without us having to worry about it. But again, all of these systems, the galls, the planarian faces, the frog face, the eyes, all of these were developed by evolution. They have millions of years of history behind them. But what happens when this continuum, so we all were a single cell at one point, so there's like the normal human here stands at the middle of this continuum, but now there's another continuum here, which is that both through technological changes and biological changes, we are already now creating novel beings that have never been here before. So if you make a novel being that has never been here before and doesn't have a history of selection, what are the patterns that they're going to have? Patterns of behavior, patterns of morphology, patterns of physiology. What are their goals going to be? What degree of persuadability? Where on that spectrum are they going to be? What specific motivations are they going to have? This is really critical. And this is really, I see this as much more important than the current issue about language models and AIs and what their status is. Forget all that. This is where the hardest thing is going to come from. Because at some point your neighbor is going to have, you know, either 49 or 51% of their brain replaced or engineered by some weird thing, and you don't want to be in a position to try to decide whether they're really human or not, right? That's not going to end well. So we really need to understand what are we actually asking here. We're not trying to use these very brittle categories. We're actually trying to understand how to predict and ethically relate to new kinds of life that haven't been here before. So what's a model system that we can use for this? And I want to show you two things that we've developed. The first we call Xenobots, and they come about when you liberate the cells from the epithelium of a frog. So here's the frog embryo. The cells up here are epithelial cells. We take them off. We put them in a dish. They don't die. They don't crawl away from each other. They form this amazing little structure. In fact, here you can see how they do it. Each one of these is a single cell. Okay, and they don't all look like a cute little horse, but occasionally you get these fun things. They sort of get together into this little pile. They move as a unit. We still don't understand how they move exactly. So it's over here. It's sort of sniffing around this other bigger area. You're going to see a little calcium flash right here, which is like a signaling event. And so eventually they pull themselves together into this kind of thing, and then the behavior begins. So then they start to swim. They have little hairs, little cilia on their surface that allow them to row through the water. They have lots of behaviors. They can go in circles. They can patrol back and forth. You can make weird shapes out of them, like these donuts. They have collective behaviors like this. They can work in a group. They can take individual journeys. They can sort of hang out doing nothing. One of the most amazing things they do is called kinematic replication. If you give them a pile of loose epithelial cells, the Xenobots will go around and they will collect those cells into little piles. The piles then mature into the next generation of Xenobots, and guess what they do? They run around and make the next generation and the next. And so both on a collective level, on an individual level, this is basically von Neumann's dream. It's a machine that runs around and builds copies of itself from parts that it finds in the environment. Now, there are many amazing things. We're just beginning to scratch the surface, but one interesting thing is that when we looked at what genes do Xenobots express that are different from what those cells would have expressed in the body. So we've liberated them from the body. What new genes do they express? Well, it turns out there's hundreds of new genes. So each one of these red cells is red dots is a new gene that they express differently. And some of the genes they express have to do with hearing. And we thought that was really strange. Why are these things expressing genes that are normally related to hearing? And we said, could it possibly be that they can hear? And so we put a speaker under the dish, and we found that absolutely, when they have certain behaviors and you turn on the sound, their behavior changes. They can, in fact, hear sound stimuli in the water. They don't do this. Embryos that they come from don't do this. This is a new thing that they've started doing. So when we talk about motivations and forms of behavior and so on, it's really interesting to ask. Now, the Xenobots apparently now care about sound. They didn't in their natural embodiment. Where do all these motivations come from? This is a really interesting, set of questions. Now, that was frog embryo cells, and you might think, well, amphibians are very plastic. Embryonic cells are certainly very, very plastic. Maybe that's some sort of frog-specific weird thing. Well, what would your cells do? If we liberated your cells from the influence of the rest of your body, what would they do? And so I'll show you this. This is, it looks like something we got off the bottom of a pond somewhere, some sort of primitive organism. But actually, if you were to sequence the genome here, you would see 100% Homo sapiens. These are perfectly normal adult human cells taken from patients who donate tracheal epithelia for airway biopsies and so on. We buy the cells. We put them in a matrix where they simply self-organize. I mean, much like with the Xenobots, we're not doing very much to them at all. The environment's the same. There are no synthetic circuits here. There are no genomic editing, no scaffolds, no nanomaterials, no weird drugs. This is basically just liberating the plasticity of these systems where these used to be part of a normal trachea. Now they're this like amazing little self-motile creature. And you could never guess this from looking at a human genome. You would have no idea that they're capable of doing this, nor could you guess what their behaviors are. What are their behaviors? Well, here's one example. If we take a bunch of human neurons and we plate them in a dish like this, and we take a scalpel, make a big scratch through it, here, this big wound, the anthrobots, that's what we call these motile structures, we call them anthrobots, they will sort of drive down the scratch, then they pick a spot to settle in. They settle in a group, so this is probably 8 to 10 anthrobots collected together into this cluster. And then what they start doing is they start knitting the neurons across the gap. They start repairing the damage. If you lift it up, this is what you see right where they were sitting here. This is, they start to knit. I mean, no one knew this was going to happen. No one had any idea that your tracheal epithelial cells, which sit there quietly for decades in your lungs, dealing with mucus and air particles and so on, have the ability to form a little creature that's going to run around and heal neural defects. And this is just the first thing we looked for. Who knows? There's probably a million other things that they do that we're investigating. But this was the first thing. So these kind of, again, I call this an intrinsic motivation because we didn't have to teach them to do this. They were not selected for this because this has never happened before. There's never been any anthrobots. They were not trained to do this. They were not designed to do this. They were not engineered to do this. In fact, like the Xenobots, these guys have 9,000 differently expressed genes, so about half the genome. So their transcriptome is completely different. So the gene in transcriptional space, they have gone to a completely different location, okay, than they would have as human airway tissue. Again, their genome is standard. We haven't touched the genome, completely the same. You would have no idea from sequencing them that you're not looking at human airway, or in fact human cells at all. They have 4 different types of motile behavior. We can draw a little behavioral diagram that shows us the transition probabilities between their different behaviors. Why are there 4? Why are there not 10 or 1 or 0? There's never been any anthrobots. There's never been selection to be a good anthrobot. Where do these things come from? And in fact, if we ask in this case, like in Xenopus, okay, the frog genome learned over the years that it's been on Earth to do these developmental stages and then eventually make these tadpoles. But also they can make Xenobots, and this is an 83-day-old Xenobot. Who knows what the heck it's turning into? We have no idea. And different behaviors such as kinematic self-replication and so on. We know when the computational cost was paid to design this. It was paid during the eons that the frog genome was bashing against the environment. That's the standard story. And that kind of makes sense. When did we pay the computational cost for this? There's never been any Xenobots. There's never been any selection to be a good Xenobot. These kinds of novel behaviors, and we still, I haven't made any claims about the cognitive capacity of Xenobots. I haven't said what their level of intelligence is. We're testing that now. We know they can learn. We're testing all of these things, so stay tuned for that. But nevertheless, there are forms of gene expression, forms of physiology of behavior in these kinds of synthetic creatures that you cannot tell an evolutionary story from. You could say that, well, it learned to do this at the same time it was doing this, but that kind of undermines the whole point of evolutionary theory, because evolution was supposed to show you a very tight causal connection with great specificity between what you have now and the environment of selection that got you here. And if you can say, yeah, the environment of selection was for this, but also it somehow got this, that isn't really how these things are supposed to work in a predictive fashion. So it's very interesting. Where do these extra things come from? Now, you might think that, okay, biology is certainly special. In fact, maybe what makes biology different than physics, than engineering, than machines is precisely this, that they have some sort of special, some sort of a special ability to have emergent, and I'll talk about this in a minute. I don't like that word at all, but some sort of emergent motivations that you couldn't have guessed from their past history, and maybe that's why biology, maybe that's the secret sauce of biology. Well, I don't have time to go into great detail, but I'll just point out, and you can take a look at this paper. The computer scientists among you can take a look at this paper. We decided to address this issue, because I don't believe that that this is a rare or that this is a property of only of very complex living systems. For maximum shock value, I wanted to take the simplest, most deterministic, most familiar thing that we could, and so we took sorting algorithms like bubble sort, selection sort, that kind of thing. Just a few lines of code, completely deterministic, totally transparent, unlike the biology where there's more mechanism, there might be quantum biology, whatever. In this case, you know exactly what the algorithm is. There's nowhere to hide. There are no more steps. You see all the steps. We found out that even something like that, there are two very interesting things about it, and there's more coming on this year. There'll be a couple more papers on this kind of thing. First of all, we found that it has behavioral competencies that are not in the algorithm. For example, one of the things it knows how to do is delayed gratification. And so as it's sorting the data, if you actually break some of the numbers so that they don't move, the algorithm says swap the 4 and the 5, but the 5 is broken, it doesn't move. The algorithm has in the standard algorithm, you assume the hardware is reliable. There are no provisions to say, okay, I wanted to swap, but what do I do if they don't swap? There's nothing like that in the algorithm. You assume that everything will go well. Well, it turns out that in that case, what it does is it sorts the algorithm, the array a little bit to move things around that stuck number, and then eventually it gets to where it needs to be. So any behavioral scientist, if you didn't tell them that this came from a simple algorithm, they would look at this and say, oh, we know what this is. This is delayed gratification. This is going backwards away from your goal in order to recoup bigger gains later on, right? But it's not in the algorithm. There's nothing in there that tells you that it's going to do that or how it's going to do that. It also does these weird side quests, for example, clustering by algo type, where these are things that are not exactly, they're not forbidden by the algorithm, but neither are they in the algorithm. They're not prescribed by it either. They're sort of, it's sort of filling up the empty spaces in between what it has to do with other things that it does that we never asked it to do. In fact, things that have nothing to do with the sorting problem itself. So I think that it doesn't take life or cells or huge complexity to have novel novel behaviors and novel motivations that would be easily recognizable by behavioral scientists if they weren't happening in such a minimal medium. And so I think it goes all the way down. And the reason I think it can, we typically have this idea that, okay, there's biology, and biochemistry doesn't tell this whole story of the mind, and we can all sort of live with that somehow. But at least there's a corner of the world that acts exactly the way it's supposed to. We have machines, we have algorithms, and the theory of algorithms and the theory of computation is a complete theory of what boring machines might do. Many people think that those kinds of theories are not applicable to living things, and I would agree with that. They do not cover what we see in living things. But my claim is they don't even cover what simple machines are. And really, you shouldn't expect them to, because they're formal models, and nobody said that any formal model has to give you the whole story of the object itself. The formal models have limitations, and we should be very careful to not confuse those limitations with the formal with limitations of the thing itself. When Magritte was telling us this is not a pipe, this is a representation of a pipe. So we can do this, and we can say this is not a Turing machine. Okay, this is a representation of a machine that has a particular sets of capabilities and limitations, but that doesn't mean that it's a full description of what's happening. And I think for exactly the same reason that living things and their capacities are not well described by our formal models of what's going on, I think this goes all the way down. Even something as dumb as bubble sort can have novel behavioral competencies, not just complexity, not just unpredictability, not indeterminism, but novel competencies and novel goal states that are not predicted by nor made easy to discover by the standard models of computation. So I think these things go all the way down. And then the final piece of this story that I want to ask is this: okay, however far down it goes, we've now realized that we need to understand where these motivations come from. If it's not going to be a history of selection and a specific design for a specific outcome, where else do they come from? And so there are two ways to think about this. One is called Emergence, and this is by far the more popular right now. But I want to argue for something different. So first, let's just ask, especially to biologists, that seems kind of crazy. If the patterns are not coming from physics or from genetics, where could they possibly come from? Well, I just want to remind us about mathematics. So this is a very simple function in complex numbers, okay? So z cubed plus 7, and so you iterate this and you draw the Halley map and you see something like this. Or if I vary this a tiny bit at a time and put the frames together, I can make a movie that is basically a kind of flyover of some of these structures. It doesn't hurt that they look very biological. They look very organic. I mean, all these kinds of things look very organic. They come from this tiny little seed. Okay, it's not compression. There's no way you're going to compress something this complex into a thing of 6 or 7 characters. This is an index into a pre-existing pattern. It's a mathematical object, and the important thing about these mathematical objects is if you ask why does it have the shape it has, the answer is not because there was a process of selection that killed off all the things that didn't look like this. Okay, there was nothing like that. Nor is the answer, because physics. There's some aspect of physics that determines this. There is no aspect of physics that determines this. You could change all the constants at the Big Bang, and this wouldn't be any different. So now we understand there are patterns with great specificity. And in fact complexity that are not the results of either a selection history or some sort of necessity of physical laws. So what do we do with these patterns? Well, the mathematicians do something interesting. They think that these patterns come from an ordered, structured space. It's not a bunch of random grab bag of things that show up as a surprise from time to time, but actually an ordered space. One way to think about it is a kind of Platonic space. So Plato talked about something like this, a space of objects that are not determined by physics, not determined by evolution. However, they really affect both physics and biology, because biology exploits the heck out of these things. These are kind of free lunches that biology uses a lot, and we can talk about that if somebody wants to ask about examples later. In physics, also, when you start asking about the very fundamentals of why bosons do this or that, the answer eventually turns out to be, oh, because there's this mathematical object and it has these symmetry groups and that's why it's like that. So eventually if you keep asking why, both in biology and physics, eventually you end up in the math department. I think that's really important. So there are two options for thinking about this. Number one, when I ask people, biologists in particular, I say, look, here are some amazing facts about networks, about logic gates, about numbers that biology is exploiting. Why are these things like that? Why do they exist? And some people say, well, that just happens to hold in our world. It's just an amazing relationship, it's just an amazing pattern that happens to hold in the world. Now, the benefits of that view is that you get to keep a nice sparse ontology, meaning that you can be a physicalist and you can say, like, there's nothing other than the physical world. Yeah, it just has some surprising features in it that are kind of cool. And that really is where a lot of people end up when they say emergence. I mean, basically surprise. Some stuff happened. It was surprising. It was pretty cool. We can write it down in our book of Emergent. Emergent facts, and that'll be that. I really don't like that view. I think it's a kind of Mysterian view that prevents a lot of research. Instead, I think we can hypothesize more optimistically that these patterns are not a random grab bag of things that just happen to hold in our world. There's a spatial structured space, a structured latent space from which these are drawn that we can investigate. And the idea is that all of these things that we make, synthetic Xenobots, biobots of all kinds, robots, cells, embryos, all of it are interfaces into that space, and thus are tools or instruments that we can use to explore that space. Now, the interesting thing is that the tools you use to explore something often determine what it is that you're going to find. In other words, there has to be a kind of impedance match between the tools and what you find. So physics tends to see mechanism, not minds, because it uses low agency tools, rulers and voltmeters and things like this. If you want to see minds, you have to use other minds. You have to use complex kinds of cognitive systems to find other cognitive systems. And so I think that's very interesting, right? What we make constrains what it is that we see, because the interfaces that we create determine what patterns from that space we're gonna be able to pull out. So I suspect that Hawking asked what breathes fire into the equations. Maybe that was backwards. Maybe it's actually the equations describe certain structures of that space, and that's what breathes fire into the facts of biology and certain facts of physics and so on. And obviously, I'm not the first person to say this. Heisenberg and Whitehead basically pointed this out too. So the model at this point that I'm proposing is simply this: there is this latent space. It contains some low agency, at least probably low agency, I'm not even sure about that, but low agency patterns that whose behavior are studied by people we call mathematicians. So math is a behavioral science. It's the behavioral science of kind of low agency patterns. But that space also contains much more complex, much more active, dynamic, high agency patterns that we would recognize as kinds of minds. And so those are the other things that are inhabiting that space. And so that means that when you make new interfaces, new bodies that are different from the natural ones, so pretty much any combination of evolved material, engineered material, software is going to be a home for these ingressing patterns. And so when you make these novel kinds of embodiments, you're going to meet some new patterns you've never met before. And so now we can talk about AIs and cyborgs and hybrids and all these kinds of things. And the last thing that I'll say before I wrap up here is this: that we can go further and we can ask at much larger scale, processes like evolution, what are their intrinsic motivations? And one of the recent things we learned actually is that there's this amazing feedback spiral where in very simple chemical networks, they can learn. That's the first thing, is they can learn. Learning increases their causal emergence. It makes the system more real, more than the sum of its parts. Forgetting does not erase those gains. And once you have those gains, you get better at learning. So it becomes a ratchet. It becomes a feedback cycle that basically points upwards towards intelligence. It's a free gift from the mathematics of networks and causal emergence. You don't need selection for this. It predates selection. So maybe these kinds of things, even processes like evolution, have a fundamental arrow towards higher intelligence that are basically they're patterns of mathematics that underlie this. And then evolution, of course, will optimize the heck out of it. But in the meantime, it comes from math. So I'll just end here and summarize that I think patterns of all kinds of spaces are ubiquitous. These patterns serve as goals for agents, very simple agents or more complex agents. Genetics, physics, emergence, these are all insufficient. What we really need to do is to map out the latent space. And an important claim is that even very minimal systems enable the ingressions of these patterns that are that take us beyond concepts like complexity, randomness, and determinism. There are other things that are really critical. And so here are some hypotheses and speculations that basically I think that everything from machines, cells, embryos, swarms, robots, whatever, are pointers into this space. Okay, they're interfaces through which these patterns ingress into the physical world. I think physics is what we call systems that are constrained by these patterns, but biology is what we call when systems exploit these patterns and use them as a kind of a free lunch to advance. I think that all engineering is partly reverse engineering, and the attitude towards your constructions and towards AI and everything else should be less Skinner and more Piaget. You want to not just have reward functions and things like that, but you want to understand what are the intrinsic motivations of the system and to what extent can we collaborate, cooperate with it, and so on, because you're going to get surprises. Not just perverse instantiation, not just complexity and unpredictability. You're going to get the emergence of cognitive properties. And so there are some other things we could say about the relationship of minds to brains along this model and free will and so on. We can talk about that. So we have a research program that we're undertaking. That's the nice thing about all this is that it's very actionable. So we're building new interfaces. We're trying to quantify the free lunch aspects. We're asking about whether you can get compute in that space. How these patterns are ingressing through the interfaces. There are many different kinds of interesting research questions that my group is now asking. So I'll stop here by thanking the people who did the work that I showed you today. We have lots of amazing collaborators in our center and beyond. There are lots of people who have funded aspects of this work over the years, and so I'm very grateful to them. In particular, there are 3 companies that have to do a disclosure, so that there's been some commercial interest in some of this work. So here are 3 companies that have licensed some of these ideas. I am not claiming that the people here or any of these people specifically endorse my model. It's a very unconventional, at least today, not the way most people think about this, but. I think it helps us to move forward. And again, all the credit goes to the various model systems, living or not, that are teaching us about these things. So I will stop here.